So the next presentation is kind of part two of the presentation that I gave. It's that ground redberry, juniper, and urea DDG-based supplements do not adversely affect lamb, rumen, microbial communities.
So I'm Sue Ishaq, and thank you so much for coming to my presentation today. Is it actually working? That's all right. So as we've been hearing recently, redberry juniper is of concern, especially in the southwest of the U.S. right now. It's an evergreen conifer, which is native to the southwestern North America. And around the last 200 years, it's moved from its rocky slopes, where it's been fine, down to our grasslands, which of course ranchers and producers are using. And unfortunately, it does reduce grassland yield and that carrying capacity. It's also considered to be a low-quality forage, mostly because it contains secondary compounds such as terpenes and essential oils such as juniper berry oil. Now these essential oils and terpenes have a possible antimicrobial action depending on the microbe and the oil in question. They do tend to reduce the palatability of this forage, and they can cause damage to ruminant tissues, especially the liver, if they're not detoxified in that rumen. So it is a big concern. However, recent work has shown that drying and grinding of juniper can actually increase that palatability and may also destroy these monoterpenes, which is what we're really concerned about before ingestion of the animal. So there is a lot of interest recently in feeding juniper to livestock, mostly to get rid of it. It's a great cheap source of feed, as Travis was mentioning before. It may possibly reduce methanogenesis, which we haven't explored in this particular study, and it has moderate nutritive value, so it's worth looking into. So our research questions, especially for the second half of this, was can juniper be used as a low-cost source of forage without negatively impacting production? Travis mentioned in his previous discussion lamb health, and then I'll be presenting lamb rumen parameters and bacterial diversity. And the second question, if bacterial diversity is actually altered, can we associate it with a specific factor, such as change in rumen pH or concentration of juniper? So as we said, we had 48 Rambouillet ewe lambs, which were about 8 months old. We had 6 lambs per treatment. These lambs were individually penned and fed ad libitum a basal diet of ground sorghum and Sudan grass hay, and then one of 8 dietary treatments, which was mostly based of distillers' dried grains with solubles, and then one of 4 concentrations of ground juniper, 15, 30, 45, or 60% of dry matter, or one of 2 concentrations of urea, 1 or 3% of dry matter. And then most of my data comes from that 34-day ruminal fluid collection via gastric tube. From there, we sequenced the 16S rRNA gene, specifically the V3 to V4 region. We amplified that with the primers 341F and 806R, and we sequenced it on an Illumina MySeq. And this is where I came in. I did my analysis primarily using mother. I quality culled sequences which were not the right length, which contained any ambiguous bases which could not be called, homopolymer runs along a certain length, anything that was identified as chimeric or not existing in reality, and anything which did not align or classify to a bacterial Silva 16S reference, basically anything that was junk or low quality, I got rid of. And then we subsampled to 5,000 sequences per sample in order to do our statistical analysis, which was mostly done using mother, R, and primer. So as we can see from the bar graph shown, that bacterial diversity, which is shown here at the family level, was not dramatically altered, and I'll get into that in a second. Treatment, as it turns out, did not have an effect on the whole phylogenetic tree structure. So overall, it really wasn't changing our mass rumen diversity. And we saw that based on our unweighted unifrac value, which looks at the structure of the tree, and then our weighted unifrac value, which is based on structure and abundance of who's there and how many of them. And we had fairly weak values for that. And then more nonsense, treatment did not affect genus-level operational taxonomic unit according to analysis of similarity, analysis of molecular variance, and then also Permanova. So really, we're not seeing huge overall changes, which you can sort of get at from this chart, which has a lot going on. So I've isolated some of the families which were of interest and which had the most dramatic changes. On the left, we have 3 families from the Bacteroidetes phylum, and on the right, we have 2 families from the Firmicutes phylum. As we can see, it's not actually even plugged in anymore. Acetaminococcus over here, ruminococcus and lacnospiraceae all either significantly increased or had a tendency trending to increase as juniper concentration increased, but it was only at the low level of 1% urea dry matter. We didn't see that change reflected in the 3% urea dry matter. On the other hand, our BSL 11 gut group and our Prevotellinaceae family had a tendency to decrease. BSL 11 decreased at a higher level of 3% urea, and Prevotellinaceae was mostly decreasing at that lower level 1%, and it did actually pop up at the end, but again, that was only trending, it was not significant. When we took a look at our samples using a non-metric multidimensional scaling plot, as seen here, we didn't see a lot of strong clustering, so our treatment really didn't affect where these were spatially. I have treatments color-coded and then also designated by shape. So again, those J's are signifying the percent of juniper in the diet, and the U is the percent urea in the diet. And what we're seeing is it's just sort of kind of all over the place. Treatments had a really low correlation of spatial variability. Their Pearson's correlations were less than 0.3. On the other hand, we did see that certain species did affect sample clustering. For example, Prevotella, Butyrovibrio, and Ruminococcus all had Pearson's correlations that were greater than 0.7. Prevotella was the most abundant genus across treatment groups at 42 to 48% abundance. This is pretty standard in ruminants. However, Butyrovibrio and Ruminococcus actually were found in low abundance, so it's interesting that they did have a strong correlation, even though that sample clustering was weak because they were not very abundant. If we go even deeper and we look at certain genera that did have a change with respect to treatment, hopefully you can read all of that, we saw that we were mostly seeing a change with percent dry matter of juniper and not necessarily with changes in urea. All the changes we saw were at that 1%. So again, up here, we've got Prevotella, which decreased slightly with increasing juniper concentrations. Again, this was trending. Is not entirely significant. Our RC9 gut group actually didn't change significantly over time, and that's another fairly important genus of interest in ruminants. However, we did see that Succinoclassicum, down here in orange, Ruminococcus and Butyrovibrio, both unfortunately in blue, all increased with increasing juniper concentrations, but again, just at that 1% dry matter urea. We did also see other genre changes, but they were such low abundance as to make them sort of not significant for this talk. If we take a step back and we look at our volatile fatty acid profile as well as our pH and ammonia, we can see that on the primary axis we have percent volatile fatty acids, and there are also tiny standard error mean bars. And on the secondary axis, shown in these line graphs, we have pH and then ammonia nitrogen in milligrams per deciliter. Overall, total volatile fatty acids, which is actually not shown on the graph, was unchanged with diet treatment, and so was ammonia nitrogen. However, when we looked at just juniper dry matter in the diet, and when it increased, we find that our rumen pH is increasing, our acetic acid percentage as a whole is increasing, propionic acid was more or less unchanged, and our butyric acid as a percentage of the whole was decreasing. When we look at when just urea dry matter was increased from 1 to 3% in the diet, we actually didn't see a significant effect on volatile fatty acids, and there was also no effect on pH. If we go ahead and mesh those things together, we find that Prevotella, as shown in this green bar here, was positively associated with both average daily gain and gain to feed ratio or efficiency. Positive correlations are shown in blue, so you can see that that light blue was, again, just a slight positive correlation, but still there. And only the significant correlations are shown. Whereas Firmicutes, shown down here in red, was negatively associated with not only urea percent dry matter, but also serum urea nitrogen, rumen ammonia, total volatile fatty acids, total acetate, and total propionate. So it did not enjoy that diet at all. And what was interesting is that body weight did not significantly associate with any specific taxa, which is too bad. And again, we did have associations with many lower abundance taxa, but there was way too much to show on the screen. So in the future. We also did see that certain genera were associated with other genera. Prevotella, shown here in this red bar here, was negatively associated with Ruminococcus, Butyrovibrio, and Succinate classicum, as well as, again, several other lower abundance taxa, which are not shown. And Ruminococcus, shown here, Butyrovibrio, just underneath, and Succinate classicum, shown vertically, were all positively associated with one another. The interesting part is that none of these genera were correlated with dietary treatment, pH, or other rumen factors, which was on the previous slide, which really indicates that there's some sort of biotic interaction going on and that these guys are deriving their own abundance. So our conclusions were that we did see small alterations in bacterial diversity, which occurred with increasing redberry juniper and or urea in the diet, mostly with that redberry juniper. However, they didn't greatly affect rumen functionality or land performance, and they certainly didn't greatly affect rumen diversity. Again, this was a fairly short-term study. Their feeding trial was 34 days plus their run-in period. So it may be that over a longer period of time we would see that. And again, we only looked at bacteria, so it would be very interesting to look at protozoa, which tend to be a lot more sensitive to changes in the diet, and see what they're doing with respect to juniper. Prevotella was decreased by both juniper and urea. It may actually be responsible for the associated reduction in body weight that Travis presented on earlier. Prevotella, the genre, digests a really, really wide variety of substrates, including cellulose and hemicellulose, depending on which specific one you're talking about. So this isn't really a surprise that killing off most of your protozoa or your Prevotella would reduce your body weight. Ruminococcus, Butyrovibrio, and Succinate classicum were all increased with juniper supplementation. Come back. Ruminococcus and Butyrovibrio have both been shown to produce succinate, and Succinate classicum is so named because it ferments succinate to propionate. Propionate was not significantly altered in the present study, and I don't believe we actually measured succinate, but that would explain why Succinate classicum might be increased in the present study. However, that doesn't answer what might have caused Ruminococcus and Butyrovibrio to increase. Is it that they were competing with Prevotella, and if Prevotella is reduced, they're able to take over? Is there some other interaction going on? And then I didn't present any of the data here, but there were actually certain genera which were discriminatory to being in that high urea diet. Some of those included Phragobacterium, Oribacterium, and Permitobacter. Again, these were all discriminatory based on LDA analysis. They've all previously been shown in other studies to be negative for urea hydrolysis. So it might be a coincidence that they're discriminatory to our diet, or it's possible that the urea hydrolysis is species or strain specific. So there was a lot more information that I didn't include. So if you do have any further questions or would like to know more, we should both have publications coming out by the end of the year. And you can contact me all over social media. And I would like to acknowledge that this study was funded by the Food and Fibers Research Grant Program by Texas Department of Agriculture. And we'd also like to thank Poet Nutrition for donating some of that distiller's grain. With that, I'll take some questions. Don't be shy.
Travis, when did you sample the rumen fluid post-feeding?
Day 34, I think we did it at 4 hours of feeding.
Do you think that there would be any change in their urealytic populations if it was closer post-feeding than before?
Yeah, well, in terms of diversity, probably not with bacteria, unless there were bacteria which were so sensitive along their cell wall that they were actually getting lysed by urea or other breakdown products or things like that. In terms of functionality, certainly yes. If we had looked at RNA transcripts instead of just bacterial diversity using DNA, we could see not only who was there, but what was going on, what's being upregulated, what's being downregulated. So certainly we would see a huge impact based on time, because that's fairly time sensitive per bacteria. And of course, after the initial feed and you've got all these breakdown products, then you've got a second surge of whoever's using those breakdown products. And certainly if we had looked at protozoa also, because they tend to follow a very diurnal pattern and tend to increase diversity based on food as well, depending on whether they're cellulitic or bacteriolytic.
Just a side, go ahead.
So the luminal fluid, that you took from the group of animals which were fed versus not fed, you pulled that or individually?
They were run individually. Yep.
I was just going to throw out that we stomach tube these, like we did in risk study, so liquid versus particle-associated bacteria. I know John Ivy has looked at that and shown some not so big differences between liquid and particle. So again, we're taking that liquid-associated bacteria mainly. We take a little plug of feed and shake it and try to get some of that off, but that's something we'll address in the paper.
Thank you very much.